Drugs and Forensic Toxicology, Lecture 2, Forensic Toxicology. So we're going to discuss alcohol, which is a sedative, hypnotic, and belongs in the category of sedative, hypnotic, and anxiolytic. Ethanol is a relatively small and somewhat neutrally charged particle. It's highly miscible with water and many organic solvents, including acetic acid and acetone. Alcohol could readily pass through the blood-brain barrier because of its high oil-to-water partition coefficient, but this partition coefficient is low enough so that it's not fat-soluble. This means that due to the low fat solubility, it results in females having higher plasma concentration than males because females have more body fat proportionally than males. In addition, the onset of action is quick and the duration is rather short, and it is metabolized at a fairly constant rate, zero-order kinetics, and the first pass of metabolism mostly occurs at the upper GI tract. When we talk about blood alcohol level, we're talking about the degree of intoxication, which is in grams percent. This is equal to the number of grams of alcohol in every 100 mils of blood. And so the legal limit for intoxication is 0.08, and 0.4 is typically the lethal limit, lethal level. And what they test for alcohol is the breathalyzer test, because alcohol passes unchanged from the lungs, from the bloodstream to the lungs into the air sacs. So the breath test is related directly to the blood alcohol level. And this is information on the breathalyzer test. You could actually read all of this information is in the textbook, or you could pause the lecture at this point and you could read over it yourself. Another test is something we can talk quite a bit, which is gas chromatography to determine the blood alcohol level. And then at site there's the field sobriety test, where you stand on one foot, you stand with your eyes closed, and you walk on a straight line. Okay. So we're going to discuss poisons. Poisons are substances, well, all substances are poisons, as according to Paracelsus. There's none which is not. The right dose differentiates a poison from a remedy. So it's a substance, when you're taken in sufficient amounts, can cause a harmful or deadly reaction. So poison, when you measure toxicity, you measure the LD50, or the lethal dose. This is the amount of substance that kills half the test population, usually within 4 hours. And it's measured in milligrams or mgs of substance per kilogram kgs of body weight. So they measure the toxicity and scale the amounts to different size animals, though estimating the lethal doses in humans is based on those of lab mice, rabbits, or rats is very uncertain because the resistance can be different between species. For example, nicotine kills humans at 0.9 mg per kg, while kills dogs in 9.2 mg per kg, pigeons in 75 mg per kg, and rats in 53 mg per kg. So here they are the LD50 of the rat and the oral dose, and it's the correlation to 50 pounds of adult human. So if it's less than 1 mg per kg, this is about a taste to a drop, and the toxicity is extremely toxic. And as you can see, it goes up to relatively harmless, and these are some some different materials. As you can see here, caffeine has an LD50 of 192. Arsenic chloride has an LD50 of 48, and you can see ricin, dioxin, and botulinum toxin has very low LD50s, and these are extremely toxic. And the CDC classifies these different poisons as biotoxins, and we'll go into a couple of these that are important for you guys to know. Biotoxins, blister agents or vesicants, blood agents, caustics or acids. choking lung pulmonary agents, incapacitating agents, long-acting anticoagulants, vomiting agents, toxic alcohols, riot control agents, tear gas, organic solvents, nerve agents, and metals. So biotoxins are a substance that causes injury to health of a living thing on contact or absorption, and it's produced by living cells or organisms. And these are typically lethal in small quantities. So they would come from plants, animal, fungus, or microorganisms. So they vary in purpose and mechanism, and they could be very highly complex or just really small proteins. And bacterial toxins, there's two types. Exotoxins, those are ones that are made and secreted by bacteria. Endotoxin, which is within the bacteria, but then it's released by when the bacterial cell lyses. There's animal toxins that are divided into two groups. There's neurotoxins, and these are the ones that act on nerve cells. And then cytotoxins, these are toxic agents to cells, and they kill cells. And the primary functions of these different biotoxins is, predation. So spiders, snakes, scorpion, jellyfish, and wasp use it to hunt. Or defense, as in the case of bees, ant, honeybee, wasp, and poison dart frog. So one of the most extremely lethal toxin available or known is the botulinum toxin. It is produced by the bacterium Clostridium botulinum, and it's a neurotoxin. And it causes intoxication, paralytic illness. So the paralysis, typically starts in the muscle of the face and then spreads towards the limbs. And then in very severe forms, it leads to paralysis of the breathing muscles and causes respiratory failure. And this neurotoxin inhibits acetylcholine release, so the toxin interferes with nerve impulses and causes paralysis of the muscles. And it's been widely used in a variety of medical uses, such as Botox, which is used to treat muscle spasms and remove wrinkles. The biotoxin anthrax, it comes from the Bacillus anthracis. It's a rod-shaped gram-positive aerobic bacterium, and it's normally present in endosperm. So since it's present in endosperm, it could exist for several years as an endosperm, and then once it is agitated, then they become an active form. They produce the anthrax toxin, which is an exotoxin. It causes edema, and it's quite a lethal toxin. And it causes chest edema, and it also leads to cardiovascular shock. So when they had to clean the anthrax during their anthrax attacks, they had to use oxidizing agents such as peroxide and ethylene oxide and chlorine dioxide and liquid bleach. So because these agents destroyed the endosperm. The biotoxin ricin is a protein toxin that is extracted from castor beans, the Ricinus communis. Ricin is poisonous if inhaled, ingested, or injected, and it inhibits protein synthesis through inhalation. You'll have respiratory distress within a few hours, and then if through inhalation or ingestion, you'll exhibit vomiting, diarrhea, and severe dehydration. The scary thing is it's easily purified from castor oil. It's a manufacturing waste from producing castor oil. And every year there's a ricin incident in the news, so it's a fairly common biotoxin. Another biotoxin is atropine. Atropines are alkaloids, these are naturally occurring chemical compounds containing the basic nitrogen atoms. So these are extracted from the deadly nightshade, Atropa belladonna, and as well as other things, and mandrake, gypsum weed, and they're competitive antagonists for the muscarinic acetylcholine receptor. So they're classified as anticholinergic drugs, and they block these nerve cell transmission, largely in the parasympathetic nervous system, and they increase heart rate, they dilate pupils, and they could produce difficulty breathing. And it is believed that these atropine extracts were used by Cleopatra to dilate her pupils in hope that she would appear more alluring. And in the Renaissance, women used the juice of the Atropa belladonna to enlarge the pupils of their eyes for cosmetic reasons. So belladonna is Italian for beautiful lady. Blood agents are ones that affect the body by being absorbed into the blood. We have carbon monoxide, cyanide, and sodium monofluoroacetate. And so we have organic toxicants, which are, in this case, we have cyanide, which is negatively charged ions, and blocks cytochrome C oxidase in cellular respiration and leads to lactic acidosis. So because our brain has a high metabolic rate, it is disproportionately vulnerable to cyanide. And you are exposed to cyanide through inhalation, you can swallow, and it's absorbed. And some of the symptoms include rapid, fast breathing, dizziness, weakness, headache, nausea and vomiting, and rapid heartbeat. And due to lack of cellular respiration, you cannot produce energy, and so on and so forth. Some gaseous and volatile poisons. These are ones that turn into gas at room temperature, and then they produce a toxic vapor. So one is carbon monoxide. So this is produced by burning fossil fuels, and since carbon monoxide binds to the same binding site on hemoglobin as oxygen, and it binds stronger than oxygen, so in a room the victims asphyxiate. Since carbon monoxide is colorless and odorless, they don't know, they become sleepy due to lack of oxygen. The brain lose consciousness, and then what it does, it turns blood like this cherry red or bright red, and then so you can see this bright red skin as a sign of carbon monoxide poisoning. Some nerve agents, these are there's a class of phosphorus or containing organic chemicals, organophosphates. These block acetylcholinesterase, so you lose respiratory muscle control, and then that subsequently leads to asphyxiation. It could be inhaled or absorbed through the skin. So the G agents are named after the German agents, or German scientists, and one of them is sarin, and it produces sarin gas when it mixes with water, and then there's also V agents, and what it does is it causes the victim to asphyxiate and then become comatose, and they suffocate. And this is in the train attacks that happened in Japan. There's also blister agents. These are ones that cause blisters to the eyes, respiratory tract, and skin on contact. So the odor resembles like a mustard plant, garlic, or horseradish. It's lipophilic. So you rarely suffer the symptoms immediately, and then so when you've been exposed to a mustard contaminated area, you may appear normal, but then within 24 hours you start developing this intense itching and skin irritation, which gradually turns into like these large blisters filled with yellow fluid. There's metalloid poisons. These are they include arsenic and antimony, and in addition, these interfere with cellular respiration, and they were typically used as a popular poison Napoleon's thought to be poisoned with arsenic by his British captors. People would die because it was in arsenic was used in weed killers and insecticides, and it was actually found. You could find traces of it in hair, nails, and bone if someone's been exposed to arsenic. And I guess when people poison, you add it to like food, and it produces symptoms that are similar to dysentery and cholera. But and there's metal poisons such as mercury, lead, lithium, and thallium. Mercury, you take it in low doses, but then it builds up after a while. So, such as like the mercury you ingest, like the fish from fish that's been exposed to mercury, and these symptoms are similar to metalloid poisons. There's choking, lung, and pulmonary agents. These cause like severe irritation or swelling of the respiratory tract, and then it causes this fluid to build up in the lungs. One example is chlorine gas, and then as well as phosgene, which causes massive pulmonary edema. Now we're going to go into forensic drug analytical methods. So the analytical methods of drug analysis and toxicologists are the same. They use the same tools. So for postmortem drug testing, this consists of a death investigation with the goal in establishing whether drug was the cause or a contributing factor in the death. So there's a lot of many fatalities that are due to accidental or deliberate drug overdose, and a small percentage is actually due to homicides. So what a forensic pathologist would do is they use this to determine what caused death and the manner of death, whether it was accidental, suicidal, or homicidal. And then the forensic toxicologist provides a comprehensive analysis of the wide variety of toxins. There's also workplace drug testing, so you evaluate body biofluids, primarily urine and blood, from employees or job applicants for drug content. And then you evaluate, and then finally you can evaluate contraband materials to identify prohibited drugs. So the specimens that are collected, so one of the first tasks is to remove and isolate drugs and other toxic agents from biological materials that are submitted as evidence. So in blood, there's a concentration of toxins that's present in blood often correlates more closely with lethal outcome than concentration in other specimens. So this is the most important specimen in postmortem toxicology. So there's two blood specimens are customarily taken. One's from the heart and the other's from some peripheral site. Urine is typically collected in pre-employment screening. It's preferred over blood because large volumes can be collected, and it doesn't involve like drawing blood from an individual. And the problem is the concentration and drug effects are the correlation between drug concentration and drug effects are poor, so you can't really tell how much drug they have taken. Could also determine if the person was actively exposed to abusable substance, and it could also be collected in postmortem investigation because there are certain toxins that are larger quantities in urine than found in blood. You take the gastric contents. This is beneficial in cases of sudden death, where with large quantities of lethal agents that are still in the stomach. So high toxic substances might have a low blood concentration but might have a high stomach concentration, and this is typically true in suicide. You could also take it from the vitreous humor because the eye is isolated. The vitreous humor is resistant to putrefaction, and you can obtain a reliable measure of a biochemical or drug in vitreous humor. At the same time when the there's been biochemical decomposition in blood compartments. And in addition, this may be the only fluid remaining in decomposed cadavers. You could take it from the bile and liver. The liver is most heavily involved in drug metabolism, so it contains significant quantities of most drugs, and sometimes could permit identification of an agent that caused death even when it's not found in blood. So since bile drains from liver, and it's rich in certain types of drugs such as opiates. Hair, which is not common, but there's drugs in hair are at low concentration, which are picograms to milligrams, and you could do a long lifetime of drugs in hair. So you could see, could estimate dates when individual was exposed to drugs, and as well as insects that have been feeding on the corpse. The majority of deaths that occur are natural deaths. So, for example, like coronary artery disease. But if you take cocaine, it could your heart rate increases, and it could lead to a heart attack, and this would be a contributing factor. So the cause of death is heart attack, while the contributing factor is cocaine. There's also accidental death, which is pretty common. It's common in children because there's dose miscalculations or mixtures of drugs, mixing prescription sedatives and alcohol. Suicide, the most common manner of death is in poisoning, is carbon monoxide, as well as prescription drugs. Homicidal poisonings are pretty uncommon, and determining poisoning to be a cause of death is actually a pretty difficult task. There's a twofold tactical approach. There's a screening and then a confirmation. So you first do a presumptive test, which is a screening test, to identify the presence of drugs and narrow the possible identity of the unknown drug. It's pretty sensitive, but it's not necessarily specific, so then you need to do a confirmatory test to show the identity of the specific drug present. Sophisticated, it's pretty labor intensive, and it's sensitive, and it's extremely specific. Many drugs fall into categories of acids or base, so you could test by controlling the pH of water solution into blood, urine, or tissues are dissolved. You can control the type of drug that is recovered. So what we have are acidic drugs include barbiturates, acetylsalicylic acid, and some basic drugs are phencyclidine, methadone, amphetamines, and cocaine. Another method is the color test. So these are chemical reactions that provide information regarding the structure of the substance being tested. So certain compounds or classes of compounds produce these distinct colors when brought into contact with various chemical agents. And so the 5 primary color test reagents are marquis, which is a reagent that turns purple in the presence of heroin, morphine, and most opium derivatives, and turns orange to brown when mixed with amphetamines and methamphetamines. There's the Dille Campagna, as well as the actually we have a figure with all of this here, so you can see it's with barbiturates, it's violet. Duquesnoy Levine, if you detect marijuana, it's purple, violet, blue. Ehrlich Van Erck, detect LSD, which is blue, purple. Scott test or cobalt diacyanate for cocaine, and you get this blue precipitate. And then afterwards, one of the microcrystalline test. It's a screening tool to confirm diagnosis with other testing methods. It's actually pretty fast, simple, and it's highly specific. So the test sample is dissolved in solution, and then the test reagent is either added to the solution or it's already dissolved within the solution that the sample is dissolved in. And the reaction between the compound of interest and the test reagent forms a solid compound that forms uniquely shaped crystals that you observe under a microscope, and it relies on comparing crystals to those that are reference standards using the same reagents. The problem is that when there's difficulties in exact matches due to there's impurities, different concentrations, and such. The immunoassays are biochemical tests that measures the concentration of a substance in a biological fluid. So it's based on using the reaction between the antibody or antibodies to its antigen. And most common immunoassays for drug testing are ELISAs, or enzyme multiplied immunoassay technique, such as enzyme-linked immunosorbent assay, or the radioimmunoassays. You could also do thin layer chromatography, which chromatography is separating and tentatively identifying components of a mixture. So you separate drugs diluted into its components, and then you could also use TLC. It uses a solid stationary phase and a moving liquid phase to separate the constituents of a mixture. And the compounds are separated by size, shape, and reactivity with the solvent. So you could test to see unknown substance and look at the characteristics with a reference sample. There's also gas chromatography, which is similar to what we just talked about with the thin layer chromatography. There's the stationary liquid phase, and then there's a moving gas phase, which is also called the carrier gas. And the stationary phase is a thin film of liquid column, and the carrier gas is chemically inert. Generally, it's nitrogen or helium. And then it's the time it takes a compound to travel from the injection port of the gas chromatography to the detector. And this is measures the relative retention time. There's also ultraviolet screening, ultraviolet spectroscopy. So the chemical substances selectively absorb some frequencies and reflects or transmits others. So we look for these selective absorption using a spectrophotometer, and it produces like this absorption spectrum. Those ultraviolet and visible spectrophotometry. So you use these to identify a class or drug group of compounds in a sample, and in this case you measure the absorbance of UV and visible light, and then you see where as a function of wavelength or frequency. And then the confirmatory test is mass spectroscopy, which uses a pattern of ions that is produced when a molecule is exposed to a beam of electrons, and then you get a chemical fingerprint. So it's typically you couple gas chromatography with mass spectroscopy, and then you get this chemical fingerprint. And there's infrared spectroscopy. This is a traditional method of confirming the identity of a controlled substance. It uses the compound's ability to absorb infrared light. So organic compounds absorb different portion of the infrared spectrum, and you get a chemical fingerprint with this as well. And so most drugs remain in the bloodstream for about 24 hours, in urine about 72 hours, and hair that's nourished by blood flowing through capillaries are located close to the hair root. And then so these drugs that are present in blood go through the capillary walls into the base of the hair, and they become permanently entrapped in hairs, hardening the protein structure. So what it does, it becomes a historical marker for delineating drug intake. Since the average human hair grows at a rate of 1 centimeter per month, you could define the timeline for drug use, and you could go back several weeks, months, and even years, depending on how long the hair is.